Okay, here we go. This is going to be ependymal cells, take 2. Here we go. All right, so next we're going to talk about ependymal cells. Okay. Ependymal cells. All right. Underline that in red. Okay. All right. Ependymal cells are also called ependymocytes. So, where is it? Astrocytes, ependymocytes. Okay. Ependymocytes. Okay. So these cells, they line your spinal cord and also the ventricular system of the brain. So the ventricular system is a series of cavities and tunnels that move cerebral spinal fluid around. Okay, so that was a lot of new words. So let's write this down and define everything. Okay, so ependymal cells, they line the spinal cord and the ventricular system of the brain, okay, in order to create ependyma. To create ependyma. Okay, so ventricular system and ependyma. Okay, so let's define these. Oh, I should have, to follow convention, should have used orange for the tangential definitions. Okay, so ventricular system. Right. Let's start with ependyma. Okay. Ependyma is tissues that line the barriers between It's tissue that lines the barriers between blood vessels, brain tissue, and cerebrospinal fluid. Okay? So this one is the tissue. And tissue just means like a large collection of cells, okay? So it goes cells, tissue, then organ system, then organism, okay? So tissue that lines the barriers between, so my shorthand for between is B/T, okay? Between blood vessels, brain tissue, and cerebrospinal fluid, which is often abbreviated to CSF. Okay, so what does this mean? Your brain is an organ, and as an organ, a living organ, it needs nutrients and it's going to generate waste. So it's going to get all of its nutrients from the blood supply. So that's why it needs to connect to the circulatory system and connect to the blood vessels, okay? So blood vessels and brain tissue, they need to connect to each other. And then when things go wrong in the brain and there's damage or if there's inflammation, but also just everyday normal waste products, that all needs to get cleaned out and washed out, transported away. That is done in the cerebrospinal fluid, okay? And actually, it happens a bit within the blood as well. The blood both brings nutrients and also takes away certain waste products. And then the cerebral spinal fluid is the connection to the immune system. And so there are a lot of protective cells in there that work to protect the brain. And then they also take out some waste products through the fluid, okay? So cerebral spinal fluid, brain tissue, blood vessels. These are all interfaces that use this tissue called ependyma, which is created by many ependymal cells, okay? All right, so now let's define the ventricular system, okay? And the ventricular system are they're the four large major cavities and tunnels in your brain. They're all interconnected with each other, okay? So they all connect. And this is where the cerebral spinal fluid is created by the ependymal cells. And it's also connected to the central canal of your spinal cord. So your spinal cord is actually hollow, and on the inside flows cerebral spinal fluid, okay? So that is the ventricular system. So four interconnected cavities or tunnels where CSF, cerebral spinal fluid, is created. And it's created by the ependymal cells, okay? And the ventricular system of the brain is connected to the central canal of the spinal cord. Okay. So now let's draw some ependymal cells so that we can better understand how they do what they do. Okay? So ependymal cells, they're a bit like boxy, okay? So they stack really like close to each other. All right? And then along one side of the cell, they have these long, thin processes. Okay? Oops, went the wrong way. All right. Okay, so here's the nucleus of each ependymal cell. They're really packed right next to each other because they're creating these barriers. They're like a cell wall almost. Okay? So these things on top, these are called cilia or microvilli. Okay? So cilia or microvilli. Okay? So once they've created the cerebral spinal fluid, they have to circulate it, right? So these long, thin, wavy things, they kind of whip them around to help circulate the cerebral spinal fluid. Okay, and then on the other side, they have little like tentacle-like feet. Okay. Okay, and this usually connects them to astrocytes because astrocytes also help create the blood-brain barrier. And so when they do that, they interface with the ependymal cells a lot. Okay, so these okay, so here's the soma. Okay, and these things are tentacle-like extensions. Tentacle-like extensions on the membrane that attach to astrocytes. Okay? So on the one side, they're facing the cerebrospinal fluid, creating it, secreting it, and circulating it. And on the other side, they're nicely anchored to the astrocytes. Okay? They are also involved, let's go back to blue. So they are also involved and because they are interfacing with the immune system through the cerebrospinal fluid, they're involved in allowing immune system cells into the brain. Okay? Involved in letting immune system cells into the brain. Okay. Lovely, lovely, lovely. Did we get everything? We defined all of our new words. Yep. Okay. All right. So our last macroglia of the central nervous system is called an oligodendrocyte. So we once again have the ending "site." Okay. So it's spelled oligodendrocyte. It's a very long name. Okay. Oh, we want red. Okay. Oligodendrocyte. There we go. So what do these do? These okay, so these guys are really cool. Their processes, their arms, actually the ends of it, they wrap around axons in the central nervous system to create myelin sheaths. So they're like the central nervous system's version of Schwann cells. So remember, in the peripheral nervous system, in our motor neurons, the typical vertebrate myelinated motor neuron, each of those myelin sheaths were made by a single Schwann cell. So it was one Schwann cell, one glia for every myelin sheath. Whereas here, the oligodendrocytes, a single oligodendrocyte can be serving, so like can be all of the little chunks all along the axon for over 50 axons, okay? Or sorry, up to 50 axons. So here we go. They coat the axons of central nervous system neurons. It's with their membrane, okay? With their membrane to create myelin sheaths. With their membrane to create myelin sheaths. Okay, so different from the peripheral nervous system. Okay, and one oligodendrocyte can serve up to 50 axons. Okay, how does this work? Let's draw it. Okay? So, here is our oligodendrocyte, and this is going to be very not to scale, okay? So, here it is, and okay, this is a highly cartoonized version of what goes on when oligodendrocytes are creating myelin sheaths. Okay, so So let's just do that as an example. This one's going to be a lazy oligodendrocyte. It's only going to serve 2 axons, okay? All right, so there it is. And this is how the neurons are in relation to it. So here's a multipolar neuron. And that's its axon. Okay, and then maybe here is another multipolar neuron. Okay, and that's its axon. Okay, so that's what they do. They literally like grab out and reach in order to protect the axons. Okay? All right, cool. So that's all the macroglia of the CNS, the central nervous system. And now we're going to really quickly talk about microglia, and then we'll be done. Okay. All right. Okay, so now we're done with macroglia. And we're going to really quickly talk about microglia. Okay, so microglia. Microglia. All right. So, microglia are between 10 and 15% of all cells found in the brain. Okay, so they're very abundant. 10 to 15% of all cells found in brain. Okay. So, what do they do? Why are they so abundant? All right, so they are much smaller than macroglia. Okay, that's why they're called microglia. They're found in all regions of the brain and spinal cord. Found in all regions of brain plus spinal cord. Okay? And they don't have a standard shape because they change shape in order to move around the nervous system. Okay? So, change shape. They're like the shapeshifter nerve cells. Well, they're glia, but glia are special. I already told you that. They change shape in order to move around the nervous system. Okay? When the brain gets damaged, they multiply because remember, they're actually a specialized immune system cell called macrophages. So they multiply when brain is damaged. Okay? And they constantly sample the environment to direct the immune system response to damage or danger in the nervous system. So they're like little field neuroscientists checking to make sure that the brain and the spinal cord are doing okay, that the entire nervous system is healthy and good. Okay? So they constantly sample the environment to direct the immune system response, to damage or danger in the nervous system. So I'm not going to draw this one because the microglia are constantly changing shape, but I would love to see what your imagination thinks microglia look like. And once again, we are still studying glia, so there's plenty of space for more discovery. All right, so that's it for major glial cell type.